Thanks for joining us for this symposium. I'm Bill Rapisi, President and CEO of the Lymphatic Education Research Network. LE&RN's mission is to fight lymphatic diseases through education, research, and advocacy. In order to win a fight, you first have to join it. So we ask, please become a supporting member of LE&RN at lymphaticnetwork.org. And we hope you enjoy today's symposium.
Big warm welcome and a hello to all of you who are joining us today. We have our guest speaker who's the leading expert on this lymphatic filariasis and elephantitis, and he has dedicated his whole career to finding a possible cure for this disease. It is my honor and privilege to introduce you to today's speaker, Dr. Ramaswamy Kalyanasundaram. Dr. Ramaswamy is Assistant Dean of Research and Head of Department at the Department of Medical Sciences at the University of Illinois, College of Medicine at Rockford. He is the Professor of Microbiology and Immunology and is Michael L. Susan M. Glasser Endowed Professor of Rural Health Professionals, Education and Research. Dr. Ramaswamy completed his veterinary medicine from India and has earned his PhD in parasite immunology from the University of Calgary. He completed his postdoctoral training in immunology from Cornell University, Ithaca. And currently, his research focuses on developing a prophylactic vaccine for lymphatic filariasis. The floor is all yours, sir.
Thank you, Dr. Rajat. It's a great pleasure to give a talk to this group. So I thought that I will give some background about what I have been doing and also where the cure for lymphatic filariasis is right now. Yeah. So with that, I'm going to share my slide. All right, so I'm going to talk about elephantiasis. It's also known as lymphatic filariasis. Brief overview of how somebody acquires the disease. Mostly through infected mosquitoes. They carry the larval stage of the parasite. When they bite, they enter through the body, reaches the lymphatic system, where they mature into adult male and female. They mate and produce an young one called microfilaria. And they are picked up by another mosquito where the microfilaria develops into larval stages, finally becoming the infective larvae. When this mosquito bites another individual, they get the infection. This is a very simple life cycle, but it's very complicated. Now, the chronic stage of this infection looks like this, where the patient develops edema in the dependent region. That's mostly the legs, where they develop enlargement due to the lymphedema, due to the lymphatic damage as well. Now, the severe lymphedema is one of the classical symptoms of the lymphatic filariasis. It is the second leading cause of disability in the world. And the disability adjusted life year, that is the year that is lost due to non-function because of the infection, is 1.63 million years. So currently, about 1.5% of the world population in 54 countries are infected with lymphatic filariasis. And another 657 million people living in these regions are threatened by the infection because the infected mosquitoes are present and they can transmit the infection. There are 4 stages of the disease. They can be classified as asymptomatic, without any symptoms, acute, where they are showing typical early symptoms of the disease, leading to the chronic disease that you can see here, and eventually leading into the elephantiasis, where there is secondary infection and complicates the lymphatic pathology. And finally, there is a group called endemic normal. These are individuals normally present in the endemic region along with the people who are infected but do not show any symptoms because they have developed natural immunity. Now, the asymptomatic patients have currently the parasite in their blood. These are the microfilaria circulates in the blood. They do not show any of the idiomatic symptoms, so they appear normal. and they don't even realize that they have the infection. So they carry this infection around so that any other mosquitoes can, when they bite, they can carry this microfilaria and transmit the infection. These patients usually sometimes secrete proteins, the blood proteins in their urine, and they also show significant immunosuppression by producing interleukin 10, which is a cytokine produced by some of the immune cells, and also the antibodies, IgG4, which is typically used for diagnosis. Now, because of the immunosuppression, generally when these individuals are vaccinated, that vaccine doesn't work. And also any of the immunomediators given to these patients does not work because of the systemic immunosuppression seen in these individuals. The acute form of the disease, this is when they start showing some clinical symptoms, mainly adenolemphangitis. This is actually inflammation of the lymph nodes. One of the typical signs is the enlargement of the lymph nodes where they are seeing the edema. And in these regions, the lymph nodes, the lymphatic system is blocked. And so that's one of the reasons the lymph fluid accumulates in the tissue. And they also exudate into the tissues, causing severe edema. That's also known as the cold edema in those individuals. And you can actually visualize the blockage of the lymphatic vessel by doing a lymphoscintigraphy. This is a method to look for the flow of the lymph in the region. For example, if it is in the leg, you can actually inject a dye and then see if the lymph is flowing. For example, in this picture on the right leg, there's a good lymph flow, but on the left leg, actually, it is blocked because of the there's no lymph flow because of the blocking by the parasite. At this time, there will be parasite causing inflammation inside the lymphatic vessel and blocking. That's how the worm looks like. And you can look at the worm by ultrasound, presence of the worm in this lymphatic edematous region, the presence of the parasite. And there will be significant inflammation. The patient may show fever and edema of the enlargement of the lymph nodes and edema in this region, lymphedema in the region. In children, especially boys, there will be hydrocele, that is enlargement of the scrotum, typically, again, blocking of the lymphatic vessels in these individuals. There could be edema in any dependable regions like the breast. In this case, you can actually see there is edema, and you can actually see the parasite inside a nest. Here's the histopathology of the lesion where the parasite is present. All these regions that you can see are the parasites. Now, by using ultrasound, you can actually see the moving parasite inside. This is known as the filarial dance. This is one of the diagnostic method, but it's expensive, and you need an experienced physician to detect this filarial dance in those regions where the parasite is present. Now, chronic lymphatic filariasis, after the edema develops and constantly all the fluid keeps accumulating in the dependent region, in this case, the leg, you can see severe edema, and also the skin starts erupting, leading to some secondary infection. So typical symptoms of chronic edema include increased dilatation of the lymphatic vessel, proliferation of endothelial cells, impaired lymph flow due to the obstruction due to parasite presence, extravasation of the lymphatic fluid, that is moving of the fluid into the tissue. The lymph flow is controlled by the valves. Unlike the regular blood flow is controlled by the heart, actually beating of the heart. In the case of lymph, because of the muscle action, there's a valve actually opens and then moves. So this valvular function is lost. And also, since it is enlarged, the smooth muscle that controls the vessel is also totally lost. And because of that, lymph flow does not happen, and all the fluid accumulates in the in the dependent region. In this case, it's a lymph. And all the lymph that has gone up starts coming down, and this is called the retrograde lymph flow that results in the accumulation of the lymph in the dependent region and results in the swelling of the extremities. And some collateral lymphatic circulation happens, but especially during the early stages, but it is absolutely not reversible after that stage is happened. You can detect again using lymphoscintigraphy to see if the lymph flow is affected. This can happen even during the acute stage, but chronic lymphedema is typical of retrograde lymph flow. Now, secondary infection is very common in this stage, and WHO has these public health workers going out to each village or station where this infection is common, and then try to clean their legs so that the secondary infection cannot happen or can be prevented. And ultrasound can be done, or lymphoscintigraphy can be done early, but at this chronic stage, nothing is visible. The worms would have already died at this phase of the chronic stage. Now, if this persists and no treatment is available for the patient, it can lead to severe damage to the skin in these individuals because of the lymph being accumulated and secondary infection that is happening in this region. So this is typically known as the elephantiasis. That's how the name came for the disease, because the leg looks like an elephant leg. Even here, extensive lymphatic collateral can occur, but even at this stage, it's absolutely not possible to reverse the pathology. There are several diagnostic approaches available. The easy thing would be to collect the blood, especially during the night, because that's the time the microfilaria come out into the blood circulation. During day, actually, they go back and settle in mostly in organs like lung and other internal organs. But during night or dusk time, they come out and circulate in the blood. You can collect those blood and look for the microfilaria in the blood. Most of the current tests available are based on detecting the IgG4. I mentioned that during immunosuppression, you have significant levels of IgG4. So there are several tests, mainly the immunochromatographic test. There are some rapid point care tests available. They're all looking for the IgG4, but they all have some some deficiency, because they are less sensitive. You could still miss some of the early infections. But the most accurate test is detection of the microfilaria or the parasite by DNA, or parasite antigen by DNA detection by PCR, which is quite expensive for patients to be done in regions where this infection is quite endemic. The current treatment for this infection, there are 4 drugs available. Diethylcarbamazine has been tried for several years since the disease has been identified, and it has been quite successful. It can completely clear the microfilaria, the circulatory microfilaria from the blood, and it has some effect on the adult parasite, but still it's not totally effective in clearing the parasite from the individual. So this is individuals in the acute stage of the infection when they are diagnosed. Other drugs like albendazole, ivermectin also has been shown to have significant effect in controlling the microfilaria from the circulation, but again has no effect on the adult parasites. So that means the drug is effective against the stage that transmits the infection to the mosquitoes. By that way, you can actually prevent the transmission. And the parasite carries some bacteria in them that is thought to be involved inducing mainly the lymphatic pathology and inflammation. So targeting, and they also are important for the fecundity or the multiple or the production of the microfilaria by the female worms. Now, if the bacteria is removed, the ability of the female worms to produce the microfilaria are reduced. By that way, you can also prevent the transmission and also can possibly kill the parasite. For that, actually, to kill this bacteria, there's a drug called, it's an antibiotic called azithromycin can be used, but unfortunately, the antibiotic course is a little bit longer than the diethyl carbamazine or albendazole or ivermectin, and also there's a need for repeated treatment with azithromycin that can cause drug resistance. Even with this, there has been some areas, parasites have developed resistance to DEC. That has been reported by one of the scientists from Canada. And for hydrocele, the only treatment is surgery. You can surgically remove that, reduce that inflammation and stitch it back so that the patient will have lesser concern about the hydrocele. Chronic cases, there's no treatment. And of course, by the time the parasite is dead and you cannot do anything, you could do probably some plastic surgery in some cases, or by adding collateral circulation for lymphatic vessel. These might help, but those are all costly treatment for individuals who live in endemic areas who cannot afford these kinds of treatments. Now, as far as cure, the global program to eliminate lymphatic filariasis was created in 1997 by the World Health Assembly. They wanted to eliminate lymphatic filariasis as a public health problem. Not eradicate, at least to eliminate the lymphatic filariasis as a public health problem. One of the major ways by which they wanted to do this is by using drugs. So the approach will be to prevent the transmission of the disease, to the mosquitoes. By that way, even infected person cannot transmit to others. By this way, you can kind of eliminate the disease in an endemic region. And this was launched in 2000. And the goal was to eliminate lymphatic filariasis by 2020. 3 drugs, diethyl carbamazine, albendazole, and ivermectin, were given to each and every individuals in an area. This includes the children under the age of 2 in certain cases, and mostly all the people, because the idea of these drugs is to remove the microfilaria from the blood of any infected individuals. As I told you earlier, the earlier stages of the infection do not show any clinical symptoms, and they still have the circulating microfilaria in their blood. So by giving these drugs, you can actually treat all those microfilaria carrying individuals. So, I mean, it doesn't cure the adult parasites, but definitely can prevent the transmission of this infection to other individuals in an area, because it's pretty difficult to do a diagnosis to find out who really has this infection in a community. For example, if there's a community of 2,000 people in a smaller village, and 1 or 2 people are infected, to search for that individual by diagnosis is probably much difficult. But if you give the medicines to all the 2,000 people, then probably you'll catch those 2 infected individuals and their microfilaria is cleared from their circulation. Now, there will be no transmission in that community. And initially, this treatment, or the MDA, it's called mass drug administration, was given annual basis. But then since they realized that that's not working, then it was changed to every 6 months, and now more frequently, maybe 3 months. As of 2020, about 9.7 billion treatments have been distributed, costing over $75 billion. In all the endemic countries, there were about 84 endemic countries, but this has been a significant improvement since they started the MDA. By 2020, the elimination was still not achieved, although significant advances has been made in eliminating the disease in several countries. The infection still persisted, unfortunately, in all the countries. This is mainly because the treatment has not been, all the individuals did not take the MDA, and due to several other reasons. So therefore, the target for eliminating the LF has been moved to 2030. So that means in another 5 more years. So recently there is an increased momentum to eliminate lymphatic filariasis because there is a lot of community awareness. People do realize that this is a serious health problem and need to be controlled. Even the village people or the endemic area people, they would definitely know that this need to be taken care of because it can damage several people in the community. People who are really infected and have severe enlargement of the leg, these are this community where they are dependent upon farming or maybe paddy field workers. So unfortunately, they couldn't go to work, and there is serious social stigma for these people, and also they cannot work. So this has significantly affected the community. So there has to be some treatment done as soon as possible. Since 2020, several communities have declared the elimination of lymphatic filariasis as a public health problem. A combination of all 3 drugs is now used with a higher frequency of treatment. Nevertheless, the medicine used in MDA have only limited effect on the adult parasites, as I mentioned. As of 2024, over 657 million people in 39 countries remain threatened by lymphatic filariasis. This is based upon the WHO report. And several cases do appear in all the countries that has been originally identified as having the lymphatic filariasis. And these were designated as hotspots. There are few individuals, so you can actually diagnose these individuals and treat them. These hotspots are reported from American Samoa, Sri Lanka, Africa, Western Pacific, India, Haiti, and Nepal. And some of these countries, this infection is slowly increasing in the number of hotspots that is appearing. One of the reasons I can explain is if any drug, for example, antibiotic, when they are given, it takes some time to reach the maximum plasma concentration. That means the drug, as soon as you take, it reaches the maximum level and then slowly declines because it is metabolized by the liver and are secreted out. Now, the amount it takes or the amount of time it takes for the drug to reach half of its concentration in the plasma is known as the half-life. So every drug that is taken has a half-life. So for in this case, in this picture, when the drug was given at 0 hour, by around 2 hours, it has reached the peak. And by around 6 hours, it has reached half the level. So there is 4 hours is the half-life of that particular drug. Now, when you consider this, for each of the drugs that was given in the MDA or the mass drug administration, the DEC has a half-life of 6 to 12 hours. That means in 6 to 12 hours, the drug will have reached half the amount concentration that is originally reached up to the plasma. Albendazole has a half-life of 8 to 12 hours. Ivermectin has a half-life of 18 hours. So that means in about a week, the effect of the drug is almost gone. So if an individual is given the drugs and part of the MDA, nearly in 1 week, the effect is gone. And if somebody did not take the medication due to not available because they did not see or available for that person, or fear of reaction, because sometimes when individuals have significant amount of microfilaria, several millions of microfilaria circulating in their blood, if they take this medication, especially DEC, that can cause allergic reaction, sometimes leading to anaphylactic shock. So within a community, when one of the person takes his medication and shows that an anaphylactic shock, the others are afraid, they don't want to take the medication. So, similarly, children are not. Mother may say that, no, I don't want to give this medication to the child. They are not interested in taking the medication because they say that I'm perfectly healthy. Why am I taking this medication? Or there are other health issues like I have diabetes, I have heart condition, so I don't want to take any of the medication. So, but if they do not take the medication, they are exposing themselves to somebody who did not take the medication, infected person who did not take the medication, and still the mosquitoes, infected mosquitoes are available in this community to transmit the infection. So in those cases, the effect of the drug, if it wanes after 1 week, even the people who are infected can get the infection. So this results probably in the hotspots. Or a second possibility is that none of these drugs have any effect on the adult parasites. So even if all the microfilaria are cleared from the blood, the female worms can recover back and start producing the microfilaria. And by probably takes 6 to 1 year to have these concentration enough to infect the mosquitoes can reach in the blood. So that could be another reason why this probably the FDA is failing. And that may be the reason why it did not work the last 20, 25 years. Another important reason that we need to do is to control of mosquitoes, the vector. So in addition to MDA, the vector control should have been an important aspect. So Sir Patrick Manson in 1887 showed that mosquitoes are very important. That's how they showed that they transmit the infection. Mosquitoes transmit the lymphatic filariasis infection. So vector control is an important tool if elimination of lymphatic filariasis has to happen. However, usually they are not included in the program because of the budget constraint. And since MDA started showing progress, the vector control was overlooked. Now, since the MDA is struggling now, there is a renewed interest in funding vector control. So it's an important component. Now, as I mentioned, there are some endemic normal. These are individuals that are naturally immune to the infection. And they don't have any microfilaria, but they have significant antibodies against the parasite, suggesting that these individuals may have been infected and somehow or other they got rid of the infection. And they have significant levels of IgG antibodies against the lymphatic filariasis. This suggested that these asymptomatic microfilariamic individuals may be a naturally immune individuals in these endemic areas. So the question that we asked is that, is it possible to develop a vaccine for lymphatic filariasis since there are some naturally immune individuals? And vaccine has been successfully used over 2 centuries to control, eliminate, and even eradicate few infectious diseases, actually. So the vaccine, in general, trains the immune system to fight against the pathogen. So I mentioned that there is IgG antibodies in these endemic normal individuals. So we collected these IgG antibodies and then tested in the laboratory to see if they can kill the larvae that is transmitted by the mosquito. Those are the L3 stage larvae. So we incubated them with the serum collected from these endemic normal individuals and then added normal cells to that one. And then we found that the larvae almost 78% of the larvae are killed, suggesting that truly those sera, the antibodies are protective. So then we depleted those IgG from the sera, and then we found that the protection is drastically reduced, suggesting that it is the protection, the IgG antibodies are giving protection against the parasite. When we replenish the serum with that depleted antibodies, the protection is regained back, again confirming that the IgG antibodies are critical for affording the protection against lymphatic filariasis. So very complicated. I will go through that in a simple, easily understandable way. So the parasite has about 30,000 genes, and the genome has already been completed. So we can express those genes on the surface of bacteriophage. These are viruses of the bacteria, and they can express each of those genes or portion of those genes on their surface. Now, we can actually use those phage to screen those sera from the endemic normal individuals that has the IgG, and then identify which antigens of the parasite induce those IgG. So if we can identify those antigens, we can use those antigens as a vaccine to create those IgG in everybody so that people will be protected against this infection. So that's exactly what we did. Using a technology, a molecular biology-based technology, we were able to screen the IgG antibodies using the cDNA library expressed on the surface of the bacteriophage, and identified several candidate antigens. And we selected those antigens that can prevent the infection entering into the skin and also the microfilaria. So it can also prevent the transmission. The MDA targets the microfilaria. So the vaccine will target the infective stage that enters into the body and also the microfilaria that's transmission. So basically, it's a prophylactic and also transmission blocking vaccine. That's the target because we have enough antigens that we identified. So we narrowed down based upon the antigens that are not present in the human, but it is critical for the parasite. By this way, we identified and narrowed down to 4 antigens that are critical for the parasite for surviving in the human and before they can establish the infection in the human. We tested each of this antigen in a mouse model and also a gerbil model to make sure that they are protective. And then we combined them so that we can get a better protection with all the 4 antigens. We call it as tetravalent vaccine. It's also, we name it as LFGuard. And we also used antigen. Generally, when you're using recombinant protein, you also need antigen adjuvant. We tested several adjuvants, and the one which we found most suitable, giving better protection, was aluminum hydroxide, which is a very common adjuvant used in all human vaccines, and also a GLA. This is an protein, an agent that promotes a inflammatory response because you need a minor inflammation for the vaccine to work at the site. So when we did in different animals, here actually I'm giving data from monkeys. Monkeys are pretty similar to human, and the monkeys do develop, once the infected monkeys do develop the typical lymphedema pathology, just like in the human. And they will have microfilaria in the circulation as well. So we vaccinated 10 monkeys. Out of the 10, we did at least 4 trials. And this is the last trial that we did. We used 10 monkeys, and out of the 10 monkeys, 7 of them did not have any infection. And out of the controls had about 3 that showed the infection, but did not show the infection. But that showed that clearly up to 70% protection can be achieved. I know that for parasitic infection, we cannot achieve 100% protection. but still 70% is really good because in the case of parasite, lymphatic filariasis do not multiply. One parasite entering into the body remains as one parasite. They produce microfilaria, millions of them, but only one parasite. So if we can remove 70 parasites, 70% parasite out of the body, the 30% probably do not cause much pathology compared to the other bacteria, virus, or even fungal infection. So we also looked at the three monkeys that were infected to see how much microfilaria. There were fewer microfilaria, suggesting that the female worms are unable to produce enough of the microfilaria, or the microfilaria produced are all killed by the vaccine, suggesting that probably this is one of the best vaccines. Unfortunately, this is the only vaccine available currently, and the controls had a high level of microfilaria in the blood. So we looked at the pathology. We kept these monkeys for almost 2 years. You can see that in the control, just like in the human. So what it is showing is that we injected dye into the leg where the infection was given. You can see that the lymphatic system is blocked. The lymph flow is blocked. So we injected the dye and looked at the dye at each time point in a lymph node. So we can actually measure how much the dye has come. So if there is lymph flow, you can actually see the dye coming up more frequently. In the controls, there was significant blockage of the lymphatic vessel, whereas in vaccinated animals, there was no block. So now we know that this can be moved forward for vaccine development. One of the first steps in the vaccine development for human is product development. This was done at Pi Life Sciences at Seattle, Washington. And they completed that study, and then they submitted everything to Nebraska CGMP facility where they prepared the bacteria, where they made the recombinant protein. So the CGMP bacterial culture was sent to University of Iowa GMP facility where they manufactured the CGMP vaccine. So now we have the vaccine already manufactured, and we have already completed the bottling. So the vials are now ready. And now we are filing IND with FDA. This is the investigative new drug. This is required for we start the clinical trial in human. So we are ready to start the clinical trial. We also tested the stability of the vaccine. This is stable for almost 6 weeks at 25 degrees centigrade. This will be perfect in endemic countries where we can take it. It also retained the protection similar to the original vaccine that we made in the lab. So this is the vaccine already ready. So we are hoping that we will start the clinical trials soon, both in the US and also in one of the endemic countries. So that's all my presentation. I have to thank several people who worked in my lab over the years. I started this journey in 2000, exactly the same time when the WHO was starting the MDA. I had Pi Life Sciences did the initial product development, and we have CGMP team completed the CGMP work, and we are completing the toxicology study with the Pacific Biolabs. And we have several people collaborated with us over the years, mostly in India, because I'm from India. So the infection is highly endemic in that nation, that country. And we also, all the monkey works are all done at Biomedical. And the study was totally funded by NIH all these years, and now also for finishing of this GMP and starting the clinical trial probably. So let me stop there, and then I will expect for some questions.
Thank you so much. Thank you so much, Dr. Ramaswamy. This was fascinating work, and it just shows the amount of work that goes in from the basic science to understanding the pathology, understanding what it takes to really form a vaccine. And it's
Thank
a
you.
fascinating journey that you took us along with to see what it takes behind the scenes of developing a vaccine. Uh, we have opened out the questions, and I see a couple of questions coming out already. The first one is, I showed symptoms over 25 years ago after visiting Barbados. I suffered swelling and skin irritation for all these years and finally got diagnosed with lymphedema. I did ask my PT to test for parasite. He ordered blood work for malaria even after I spelled out filariasis. Could I get tested to determine if this is in my system after all these years? I've never had chemo, so getting lymphedema is a mystery, and I often wondered if this is caused by this. Do you have anything to say?
Yes, unfortunately, 25 years back now, the parasites must have died in your lymphatic system. And so they will not be able to produce any microfilaria, so you may not see anything in the blood. So there's uh, you can look for some antigens by PCR, but it is too late. The parasites are all dead and gone, and the inflammation persists now.
Sure. Another question is, will the parasite expire naturally if unable to be unable mul- basically because of antibiotic treatment?
Um, Experience die, yes, naturally. The lifespan of the parasite is about 10 to 12 or maybe even 15 years maximum, but they don't live longer than that and they die. Once they die, they get disintegrated and absorbed in the system and probably eliminated. But the inflammation um, remains.
Uh, Dr. Ramaswamy, I have one question
Sure.
as we wait for other people to ask their questions. Uh, what is the sweet spot? How early can we diagnose and detect the parasite in our body so that we can basically reduce our chances of getting lymphedema?
Well, from larvae entering into the skin after the mosquito bite, the parasite takes about 4 months, probably. The microfilaria appears in the blood earliest, but the number increases as the female worm starts producing more microfilaria.
Mhm.
Unfortunately, the individuals who are infected do not show any
Uh-huh.
symptoms, so there's no reason for them to come unless somebody looks their blood under the microscope.
Mhm.
That's the difficulty. So that's why the WHO started by giving MDA to all the people because it's very difficult to identify who has the infection, who does not have. We were in India. One time I visited several villages in Maharashtra,
Yeah.
and we went through all these villages. We go in the evening, dusk time, and ask people to come. We will do the testing. So we have about 100 people lined up.
Yeah.
So we collect blood and then immediately looked into the microscope. Of course, we didn't have any light or anything. We had to give our cell phone. And I've taken those pictures, and that's what you see here. The microfilaria migrating is actually from those patients. And there were several people with enlarged limbs. And one of the individuals I showed in the picture is one of the elderly gentlemen who have two cattle. I mean, he works in the paddy field. He goes
Mhm.
out every day, and then he still walks around. So we tell him probably the parasite. We tested his blood. The parasite is already gone. So the
Oh.
same, the first lady who asked. So he didn't have any parasite at that time. So he had the infection for several years, but he
Mhm.
still walks around. But he cannot go and work in the field because it's very difficult for him to walk, painful. Um, yeah, treatment. None of the three drugs, unfortunately, kills the parasite.
Mhm.
And the parasite naturally dies in the patient.
Sure.
Some studies shows that the DEC can partially kill the parasite, but It's in several studies show that even after several doses of DEC, the parasites are still alive.
Well, that is essentially one of the biggest challenges, specifically when we talk about resource-deprived locations
Right.
like this, where the healthcare access is not as great as it is in US. Uh, like just to have you seen any cases in US for this? Because I believe maybe Florida might be one of the places we might be able to see something like that, or there is nothing.
Florida, I mean, we had the infection in North Carolina,
Oh.
but that was controlled. The only way probably to eliminate China eliminated the infection by totally removing all the waterlogged areas.
Mhm.
So for anybody from endemic country, I would strongly suggest that control all the mosquitoes around you, your
Mhm.
surroundings. I mean, some of the houses that we visited, actually, they have the cattle, the bulls staying in front of their home. They put a small shed,
Right.
and that's tons and tons of mosquitoes. We
Mhm.
collected some of those mosquitoes and then brought to the laboratory. We looked at those mosquitoes were infected. So showing that there is transmission already happening. And some of these areas has been declared by the country, India, that there's no transmission happening, but there is transmission happening there. We demonstrated that. Look here, there is mosquitoes infected. So it's called the xeno monitoring. So you can monitor the mosquitoes in the area. But the best thing is clear all the mosquitoes out of the area. And then, of course, MDA is critical. I mean, you need to give drugs to people to treat. And of course, vaccination is the only way to do that. And there is a question from
Yeah,
Valeria Mills.
I see a couple of more questions there. Like, your vaccine development for filariasis-based elephantitis is groundbreaking. I'm researching secondary lymphedema linked to lipedema and lymphatic trauma. And I wondered if your team has considered whether the same immune and regenerative mechanisms might apply for non-parasitic form of lymphatic failures. Do you have any insights on that, sir?
Unfortunately, the vaccine antigen is specific to the parasite. So the IgG antibodies the patient developed is against the parasite. And that's where they were able to kill the L3 larvae and establishment of larvae. And that's the reason why there's no parasitic establishment and no pathology. It's not the vaccine prevented the pathology, it's the vaccine prevented the infection. And by that way, no pathology. So I hope I answered.
Uh,
Um,
So, are the same immune or regenerative mechanisms that apply? You said like that's not the case because it's mostly pathogenic mechanisms are different
Correct.
to
But
the.
while this vaccine is specific to filariasis, do you see its potential use for any other diseases? In fact, the genome of lymphatic filariasis, the Wuchereria bancrofti and Brigham-Milley, some or other there is a genetic similarity or the gene similarity to other parasites, other phylarid parasites. What we found is that the Dictyocolus viviparus, this is a dog heartworm parasite, has very similar genes. And we found that the genes that we identified has a significant similarity to the dog heartworm parasite as well. So we developed a vaccine for that one as well because of that similarity. So maybe other filarial parasites that have similar genes, um, it is possible that the vaccine might work for them as well.
Sure. We
But
have
not
another
for
question.
other parasites.
Mhm. Another question is, what post-therapeutic protocols are used to manage persistent edema and fibrotic tissue remodeling? Specifically, have there been measurable improvement in lymphatic drainage, collagen realignment, and scar tissue following the immunological intervention?
Yeah. So I don't think that that's being done. They just help to prevent any secondary infection on the skin in these individuals. But lymphatic drainage has been tried early on, but I don't think that it is now used as a one of the treatment for that one. And you can definitely try in the early stages before it becomes chronic. There's a possibility that you can actually do all kinds of collateral circulation, remodeling of the realigning of the collagen, and reduce the formation of the scar inside the area where there is damage. It's possible, but those are all very expensive treatment in the endemic country where the people possibly the daily wages are probably less than $10, and so they cannot afford unless the government comes back and offers this treatment. If there's anybody who can afford this treatment, it's possible. And this infection is not found in city areas, or it's usually found only in rural areas. So those individuals living in these rural areas cannot afford to major treatments. So it has to be something that can be affordable to those people.
Sure. Dr. Ramaswamy, we are almost at time. I want to really thank you for this fascinating presentation. We see a lot more questions coming in. If you want to take one last question before we sign off.
So there's part of that question is that finally is the vaccine currently being implemented in African region? The dog, it's all early clinical trials going on right now. It has not reached up to the field for the dog. And for human, no, we have not even started the human clinical trial. So unless that is done, probably it may not be available. But we are trying to find a partner who can do in Africa, but right now we are targeting mostly in US and India. There's a possibility that we can do the phase 1A trial in these countries. So thank you. Yeah, sorry.
Thank you. Thank you so much, Dr. Ramaswamy. This was fascinating an informative session for all of us who have just heard of lymphatic filariasis, have never seen what the parasite looks like and how damaging it can be for the lymphatic system all itself. Thank you so much for the presentation, your precious time that you have taken out of your busy schedule. And we wish you all the very best for the vaccine trials that are going forward. And we hope that this vaccine sees the market very soon. Till next time, I'm your host, Dr. Rajit Mulay, saying goodbye and thank you so much.